Hello, everyone. Welcome to today's educational webinar by NOM. Today we have as our guest Dr. David Goldstein from Duke University. He's a professor of genetics there, but moving soon to Columbia. David's been at the forefront of human genetics for quite a long time, and his history is involved in disease genetics, pharmacogenetics, population genetics. Today he'll be talking to us about precision medicine in neurodevelopmental disease. During the talk, if you find you have questions, please go ahead and enter them in the GoToWebinar control panel. There's a section for you to enter questions, and we will address those at the end. We'll have a Q&A session so that you know we're recording this webinar, so we'll be able to share this with you again later. David, anytime you're ready, please go ahead.
Well, thanks very much, Ben. It really is a pleasure to join you and everyone at what is really one of the most exciting times, I think, in the genetics of neurodevelopmental diseases. What I would like to do in the next 45 minutes or so is tell you about a few of the studies that we are doing in the genetics of epilepsy and a few other disease areas. And I hope convince you that neurodevelopmental diseases is really poised, as a group, they are poised to join cancer in providing some of the clearest examples of where understanding the underlying molecular basis of disease points the way towards targeted therapies. I think that really is a path that we are now on, and I hope by the end of this presentation you will agree. So the first set of studies that I'd like to tell you about emerged from a project funded by the NINDS at the NIH called EPI4K. And I'd like to make clear that the work that I'm talking about is collaborative work involving a very large number of investigators involved in the EPI4K project. And overall, EPI4K is led by myself, Dan Lowenstein at UCSF, and Sam Berkovic at the University of Melbourne. And the project I'll be telling you about primarily today focused on epileptic encephalopathies, also involved substantive input from a number of people at Duke University that are pictured and named on this slide. And I'll just introduce the two epileptic encephalopathies that we have concentrated on the most. You can view these as classical epileptic encephalopathies, infantile spasms and Lennox-Gastaut syndrome. These are usually very serious epilepsies that start early in life. And before we ever got involved in the work, it was already known that the handful of genes that had already been implicated in epileptic encephalopathies usually carried de novo mutations or brand new mutations in the proband that conferred risk of disease. And so because of that, we decided to focus our initial genetic discovery efforts around finding de novo mutations in patients with these epileptic encephalopathies and trying to determine whether any of those de novo mutations could be securely, statistically implicated in risk. And so what we did is, in the last largest analysis that we performed, in this case in collaboration with another big epilepsy genetics consortium called Euroepinomics, we sequenced 356 patients with one of the encephalopathies and their parents, and we found all of the de novo mutations in the exomes or protein coding parts of the genomes of these patients. And I'm going to skip absolutely all of the methodological details on how we did that. The only comment I'll make is that there are a lot of tricks for how to do that well, to use next generation sequencing data to identify candidate de novo mutations and then to confirm those with Sanger sequencing. But if you use kind of current best practice, you can do this really well. You find virtually all the de novo mutations that are really present, and it's not too laborious in terms of the necessary Sanger sequencing confirmation. I do want to emphasize, though, that it is still critical to do the Sanger confirmation, because even when you look hard at the variants in next-gen sequence data, sometimes variants that look good aren't really there. All the results that I'm describing to you now are based on de novo mutations that have indeed been Sanger confirmed. And so just as the overall description of the results, what is shown here is the number of de novo mutations observed in the exomes of each patient, and then the number of patients that have each of those numbers of de novo mutations. And so you see that around 100 of the patients that we sequenced, we didn't identify any brand new mutations in their exomes. A similar number had just one, and so on. And overall, this is consistent with most of the previous studies showing that on average there's just over one de novo mutation per exome, and there's no evidence of variation in the burden of de novo mutations across patients. This shows a way of depicting the statistical analysis of such data. And I'm going to, again, skip the details here, but I do want to emphasize one really important high-level point. And that is that, of course, when we look at de novo mutations in patients, what we're interested in is asking, essentially, are there too many de novo mutations to be explained by chance, either overall or in particular genes in our data set? And if there are too many to be explained by chance, then you can infer that some of the mutations must be influencing risk. So that's essentially how we implicate mutations and genes in conferring risk. But when we ask that question of whether there are more than we expect, we have to predicate that on the expectation for the mutation rate, either in the genome as a whole or in particular genes. And what that means is that we cannot simply talk about whether any genes have recurrent mutation, which is unfortunately how a lot of people did think about and talk about these data until relatively recently. A set of patients would be sequenced. If there were 2 mutations observed in a gene, people would say that gene must be involved in risk. What this figure illustrates is that you can't do it that way. You need to think about the number of de novo mutations as a function of the effective mutation rate of genes. And so what I show here on the x-axis is estimating what the overall mutation rate for a gene is, given its size and the mutability of the exact sequence encoding that gene. And on the x-axis, the mutation rate is increasing towards the right. And on the y-axis is the number of de novo mutations, and the color is a heat map representation of significance for the particular combination of a certain number of de novo mutations in a gene with a certain overall mutability. And so what this shows is that if genes are bigger or more mutable, you need more de novo mutations to achieve statistical significance. And we have used a conservative approach for declaring significance for involvement of individual genes, which requires that there is an excess of de novo mutations that is sufficiently great to be unlikely, given that we are correcting for all the genes in the genome. That is, any gene in the genome might have had multiple de novo mutations in it that would impress you. And any point above the red line is significant, correcting for all the genes in the genome. And so what is shown here is that in our data set of only 300 plus individuals, we have a handful of genes that have a clearly significant excess number of de novo mutations beyond what can be accounted for under the null of none of those de novo mutations influencing risk. And so that means that we have secure statistical evidence that GABA-RB3, STXBP1, DNM1, FCN1A, that these all carry mutations that confer risk. Some of those are already known epileptic encephalopathy genes, and some of them are newly implicated epileptic encephalopathy genes in the study that we've done. I'll tell you a little bit about that in a minute. But one point to pause and really emphasize is that this really shows how well this paradigm works of using the identification of de novo mutations genome-wide to identify new disease genes. Here we're talking about looking at only 350 patients for what is a complex trait. There's no way you could argue that epileptic encephalopathies are a single gene trait. And yet, in a modest sample size, we have absolutely secure statistical evidence for multiple genes conferring risk, and some of those are entirely new genes. So that really is, I think, pretty encouraging that next generation sequencing approaches really do afford us the opportunity to make systematic progress in unlocking the genetics of these kinds of conditions. The next slide addresses what I think has been a real concern for a lot of us who have for many years felt that the most likely underlying genetic cause of many human diseases, perhaps even most, are rare mutations of at least moderate effect. The concern that many of us had that view of genetic architecture is that it could really complicate efforts to personalize treatment on the basis of underlying genetic cause. And if you think about it for a minute, to the extent that the underlying causes are rare mutations and at an extreme, every patient has a different underlying genetic cause, there's really no way we can then use genetics to organize patients into groups that are helpful in terms of treatment choices and so on, because everyone is, again, unique. And moreover, there really isn't any way we can effectively use that information to guide new drug development strategies, because it's very difficult to imagine drug development strategies being applied to single patients. And so what that means is that we need to think about whether the different underlying genetic causes organize in any sensible way that we can capitalize on. And in this regard, I think the results from the epilepsy sequencing are really very encouraging, that even though the underlying genetic causes are very rare, they do indeed organize into much more common biological processes and pathways that are affected by those underlying mutations. And I illustrate that point here by noting that of the 356 patients that we looked at, 8 of them carried mutations in GABA receptor subunits, and 12 of the 356 carried mutations in genes that encode proteins that appear to play direct roles in membrane dynamics generally, and perhaps synaptic vesicle trafficking specifically. And I think that the latter may really be emerging as a major theme in what is dysregulated in some epileptic encephalopathies. And so here are 2 of the genes that appear to play a role in synaptic vesicle trafficking, and that also are involved in epileptic encephalopathies. The already known STXBP1 gene encodes a protein involved in exocytosis, and DNM1, which we newly implicated, encodes a protein involved in endocytosis. And so this is just an illustration of the function of the DNM1 protein, which had actually been worked on already in mouse models, but had not been implicated in human epilepsies before. And it shows its central role in synaptic vesicle trafficking. So what we wanted to do is ask the question of how the mutations specifically observed in epilepsy patients influence DNM1 function. Here is a cartoon showing where 3 of the mutations that we identified in patients are found in DNM1. And this slide summarizes results that an undergraduate in the lab, Ryan DeHinza, obtained by looking at expression of these mutants in heterologous systems. And what he was able to show using a transferrin uptake assay is that these mutations have quite a dramatic impact on endocytosis. And in fact, work that was done at the Jackson Labs by Rebecca Bew-Mill and Wayne Frankel showed that in the analogous mouse model for DNM1 mutations called FITFUL, there is specifically a synaptic vesicle dysfunction where the FITFUL mouse synaptic vesicles are aberrant in both size and number. And so together, what these data show is that the human mutations have an effect on endocytosis, and it looks like from the mouse mutations that the endocytotic effect of these mutations specifically affects synaptic vesicles. And so this directly connects the mutations causing the epilepsies to synaptic vesicle trafficking, and gives us a clear target for therapeutic intervention in at least a subset of the epileptic encephalopathies. So this, to us, was really encouraging that we are beginning, as I said, to know how to make systematic progress on finding the mutations that cause disease, at least for some kinds of human diseases. And we were, in fact, so encouraged by that progress and other similar examples that we started thinking about whether it might be possible to take on the even harder challenge of making inferences about what mutations might be causing disease in individual genomes. So if you think about it for a minute, what we've been doing so far is using clinical data to organize patients for us. So we look at the presentation and we say, okay, all these patients are similar and they all have infantile spasms, or they all have Lennox-Gastaut syndrome, and then we look at those patients as a group. It's a harder problem to take an individual patient where you know something is wrong, but you don't know what other patients are like your patient. And what we wanted to know is whether we could use the evolving tools for identifying disease-causing mutations to try to make at least educated guesses about what mutations might be responsible in that more challenging setting. And what we started thinking about specifically is the idea of using what we think of as a signature of pathogenic mutations. And the one wrinkle here of what we're doing in comparison to what's been done many times before is that in getting such a signature, we combine a prioritization score for whether a variant might be harmful at the level of the variant with a prioritization score at the level of the gene. And we think it is this combination that really provides power. And specifically, the prioritization score that we use at the level of the variant is polyphen, but you could, in fact, use a variety of different approaches. And the one that we use at the level of the gene is an intolerance scoring system that we recently introduced in PLOS Genetics. This work led by a very talented postdoc in the group, Slavoj Petrovsky, and a reference to the paper is indicated there. And the basic approach is illustrated on this slide, where all this slide is showing is a simple count of the number of variants of different types that we observe in a database of exome sequences that was generated as part of an NHBLI-funded study and that now resides in the so-called exome variant server database. And what's shown on the x-axis is the total count of variants in each of the genes, and this is looking at every single sequenced gene. And on the y-axis, what's shown is the number of variants that are annotated as functional and that have a frequency in the human population of greater than 0.1%. And so what's happening here is that we use the total number of variants in the gene to predict the number of common functional variants. And this constitutes a genome-wide prediction in a standard regression framework. And then, of course, what we are interested in is genes that deviate from the prediction. And so, for example, you can see in red, we indicate those genes that are far below the line. And so these are genes where the total number of variants observed would predict that there are more functional variants than there actually are. And for such genes, we can reasonably infer that they are under negative selection. That is, a variant that does something to the protein that these genes encode, a missense change or something that changes the expression or a stop, those are selected against. And we would therefore be more impressed when we see mutations in a gene like that than one of the blue genes where there is more functional variation than expected. And the only other comment I want to make here is that the blue, of course, not only reflects the absence of negative selection, but can also reflect the presence of some kind of balance selection where functional variants are selected for, perhaps in response to pathogens. And so we give every single gene in the genome a score, and we, of course, are going to be more impressed when we see in a patient a mutation that looks nasty, so it's got a high polyphen score, and that's in a gene that doesn't tolerate genetic variation. And I also just want to emphasize that these distinctions that we are making amongst genes are not modest distinctions. They are really striking distinctions. So we can actually use this kind of an analysis just simply using control sequence data, and we can identify genes that literally never, either overall or in parts of the genes, never have certain kinds of functional variants. And what that means is when we see such a variant in a patient, we actually can infer it probably is doing something. So let me now try to convince you that that's true. What I show here is a pictorial representation of what happens when we look at de novo mutations in individuals that do not have any clinical diagnosis. So these are individuals that have been sequenced as part of, for example, autism sequencing studies, but they're the unaffected relative. So these are controls, and all the de novo mutations have been identified, and we've now looked at their characteristics. And what we find is that if we try to count up all the de novo mutations that are in intolerant genes, so as I just described the genes that are not tolerant of genetic variation in the human population, all the de novo mutations that are in such genes and that are polyphen damaging, and with a further filter, so I haven't described, but a straightforward filter, which is whether the gene is essential in the mouse. So in homozygote form, if you knock it out, the mouse doesn't survive. If we count up the number of de novo mutations that have all of those properties in controls, we find that fewer than 2% of controls have such mutations in a mouse essential gene, polyphen damaging, and a gene that's intolerant. Okay, that only happens less than 2% of the time in controls. However, if we then turn to a heterogeneous clinical population where kids are referred to a clinical geneticist for having a presumed genetic condition, but you don't know what it is, but you just think that they have something serious. If you look at that population, and we actually did this in children that came through the clinics at Duke University, not selected on any other basis except getting referred to such a clinic. When we look at those individuals that have been sequenced, we find 15% of them have mutations matching exactly the characteristics that I just described that only occur in 2% of the controls. That is a damaging mutation that occurs in a mouse essential gene that is intolerant to genetic variation in the human population. Now, this is a truly remarkable observation, and it is remarkable because what it is telling us is that when we see such a mutation in a patient, even if it is a patient that has an undiagnosed disease, you have no idea who else has something similar, you just have the one patient to work on, a classic N=1 study. When we see such a mutation, the majority of times when we see such a mutation, it itself will be contributing to that individual's disease. And that really is remarkable, and it does directly follow from this comparison. So what this is really illustrating is that already, and it's very early days for this kind of work, but already we are actually getting really good at interpreting individual patient genomes all by themselves, simply using the growing knowledge about what happens in the general human population in the genome. And I would emphasize another point here. We're already good at this, as this slide illustrates, and the key to power in this kind of an approach is the number of individuals that we have sequenced in total, because we learn much more about what parts of the genome are tolerant and intolerant as a function of the number of individuals that we've sequenced. And so we are only going to get better and better at this. So that really is pretty striking, and in fact so striking that perhaps some people need to be convinced that this really works. So let me give a couple of illustrations that you really can make useful inferences in individual patient genomes. This is a picture of Bertrand mites, and Bertrand had for many years undergone what is often referred to as the diagnostic odyssey, where he had gone from specialist to specialist as people tried to figure out what is wrong with him. And he presented with symptoms that have many similarities to disorders of glycosylation, but all of the genes that encode known disorders of glycosylation turned up negative. And he eventually got into our exome sequencing study at Duke University, in fact, one of the early patients. And when we sequenced Bertrand and his parents, what we found is that he is a compound heterozygote in a gene called N-glycanase, which at the time that we did the analysis had never been implicated in a human disease before. However, the genes that cause disease that looks a lot like what Bertrand has, those are involved in putting glycans onto proteins. And Bertrand turned out to be a compound heterozygote for a protein involved in taking glycans back off of proteins so that when the proteins are misfolded, they can be degraded. And so just in terms of the biochemistry, he had mutations in a gene that's related to genes that cause not dissimilar conditions. Moreover, we took the approach that I described before and asked whether this kind of a genotype that Bertrand has ever occurs in people that do not have a diagnosis. Not the individual mutations, let me emphasize, because that is uninformative. A lot of people say a given variant in a gene is never observed in controls. That actually doesn't matter. That's not hard to find. It's the type of variant. And so what we did first is ask, do these mutations that Bertrand is compound heterozygote for have a functional effect? And in fact, they knock out the protein almost completely. We actually showed that experimentally. And then we asked, do apparent loss of function mutations ever occur together in anybody in this gene using our own control data and all the control data we can find? And the answer was no, that never happens. Bertrand's parents are carriers, one each for these loss of function mutations, and that happens, but we never see them in combination. We put that all together, we came to a view that it was likely, though not certain, that Bertrand's condition was a brand new condition, previously undescribed, so not just a new gene, but a new condition, previously undescribed, that has a lot in common with disorders of glycosylation, but with the novel feature that when Bertrand cries, he actually doesn't make any tears. And so this was a new syndrome we suggested called N-glycanase deficiency with those characteristics on the basis of analyzing a single genome. It turns out that that inference was correct. You can read the story about how it was confirmed in the New Yorker for anyone that is interested. I refer you to this story in particular because it not only tells how the genetics works, but more importantly, it describes the role of the patient families in confirming the condition. And this is, I think, a really, really important point for us all to keep in mind going forward in this kind of work. After we got the putative diagnosis, we actually shared that with the family. And I do want to emphasize that that is not always done. It's sometimes the case that groups will only share when they are absolutely sure. We shared when it was only a possibility. But in retrospect, I think that was very much the right decision because we, of course, after we had the putative diagnosis, tried to find confirmatory cases. And we, over an 18-month period, were able to find one additional case. But the family of Bertrand, his mom and dad, Christina and Matthew Might, themselves were directly involved in searching for additional cases. And they were directly or indirectly responsible for an additional 7 cases being identified and confirmed. And so this really emphasizes to me the value of drawing patient families into these research efforts. I want to now turn to emphasize that there are real therapeutic implications of the progress that's being made in genetics. I want to also caution that this is not the rule. It is not the case that whenever you get a diagnosis, it makes a difference, a real difference to the management of the patient. In fact, it is very much a minority of the cases where getting a diagnosis really makes a big difference to the management. But it is an important enough minority that this constitutes a real motivation and, in fact, I would say a responsibility for carrying out this kind of work, and indeed, as we'll see soon, for carrying out this kind of work as early as possible. So the first example of therapeutic implication that I want to describe relates to a gene called KCNT1 encoding a potassium channel that has been implicated in two different kinds of epilepsy. One, autosomal dominant nocturnal frontal lobe epilepsy, which is pretty much exactly what it sounds like. And it is not a good epilepsy to have, but it is not as bad as some. And the other epilepsy that this gene is involved in is a early onset, very severe epilepsy characterized by migrating focal seizures, and this really is a very devastating epilepsy. Now, functional work that's been done in the Petru lab in Melbourne and other labs have shown in a classical oocyte model where the channel is expressed in an oocyte and the effect of the mutations characterized. This work has shown that the mutations that cause epilepsy are gain-of-function mutations, and specifically the mutations cause increased current mediated by this channel. And more interesting than that, there's a very clear genotype-phenotype correlation where the mutations associated with the not as bad epilepsy have a modest increase in current, approximately threefold greater than wild type. And the mutations that are found in the patients with a more severe epilepsy usually have a greater increased current, closer to 5 or 6-fold over wild type. And beyond all of that, Steve Petru specifically was able to show that all of the mutations that have been studied so far, without exception, not only have this gain of function of increased current, but they all are responsive to quinidine, whereby the administration of quinidine in this experimental setting reduces the gain of function associated with each of these patients, each of these mutations. And that suggests the possibility of a targeted therapeutic of giving patients that have KCNT1 positive epilepsy quinidine, because quinidine can undo the gain of function associated with each of these mutations. So we and others have in fact been evaluating quinidine in patients that have KCNT1 positive epilepsy. Here are two patients that we have followed at Duke that have KCNT1 positive epilepsy. Both of them had uncontrolled seizures and missed many developmental milestones. And one comment I want to make about carrying out targeted therapeutics in epilepsy is that the barrier to trying something as an off-label usage for a drug where the tox profile is relatively well known is actually very low, because when seizures are uncontrolled, it is a very appropriate risk-benefit trade-off to try a new therapy that really might improve seizure control. And this itself really does give us an opportunity for trying targeted therapeutics in epilepsy. In any event, we first determined that the mutations that the patients carry are indeed sensitive to quinidine. I think that remains critical to be sure that you are targeting, correctly targeting the treatment to the actual patient you're going to give the treatment to. And what we found in these two patients, as illustrated for one of them here, is that in one case we had a reduction in the seizure burden after the administration of quinidine, and in the other case we had a very little effect in the administration of quinidine. And I do want to emphasize that this is in fact my expectation in general, that we will probably improve treatment by these targeted approaches that emerge from simple in vitro models, but the first thing that we try will not represent the long-term cure. I think there'll be a lot of fine-tuning that we'll have to go through to get a long-term cure, but this experience does suggest to us that quinidine is a useful direction in the treatment of KCNT1 positive epilepsies. And I also want to mention that there was a similar experiment performed at the Children's Hospital of Philadelphia, and they actually did achieve seizure freedom for a KCNT1 positive patient upon the administration of quinidine. But I also want to emphasize that we have to be extremely careful in whom we try these targeted therapeutics. And a point to emphasize about quinidine is that this really is an example of precision medicine in the sense that quinidine would never be given to an epilepsy patient under any circumstances unless they were KCNT1 positive, because it is not an anti-epileptic drug, and in fact you would most certainly and appropriately get in trouble for giving quinidine to an epilepsy patient unless you knew they were KCNT1 positive. This slide emphasizes how careful we need to be in thinking about carrying out those kinds of targeted therapeutics. We had another patient at Duke that had been clinically sequenced, and the lab report came back and it highlighted a possible pathogenic mutation for the patient's epilepsy in KCNT1. And they said that it wasn't certain because only the mother was available, not the father, and it could have been inherited from the father, but it looked suspicious, it wasn't observed elsewhere, and it might be responsible. And so this patient was considered at Duke for enrollment in the quinidine trials as possibly having a KCN2 positive variant. But we, of course, evaluated that variant in the lab before we decided to proceed with enrolling that patient in a quinidine study. And what's illustrated here is the way that we look at ancestry using a standard principal component approach in the lab. This analysis was performed again by Slavoj Petrovsky. And all that's depicted here are points for different individuals that we've analyzed ourselves that specifically have either European or Middle Eastern ancestry. And the exact position of each of these points reflects the ancestry of the individuals. And I won't go into how that happens, but that works, and it's very well established, as I said. And we've highlighted in yellow the individuals that we have studied genetically that self-declare as having Ashkenazi Jewish ancestry. And we highlight in triangles the individuals in whom we have observed that exact KCNT1 variant that came back from the diagnostic lab. And so what you can see very clearly in looking at this plot is that that variant that came back in the report from the lab does not cause epilepsy. It actually is an Ashkenazi Jewish polymorphism. And because there weren't that many Ashkenazi Jews that were included in whatever reference data set the lab had looked at, they didn't recognize it as a polymorphism specific to Ashkenazi Jews, but this analysis very clearly shows that. So this really makes clear that in marching forward with these kinds of precision therapeutics, we really need to be extremely careful to get the genetics right. And we will make mistakes, no matter how careful we are, but we need to do the best job we can in order to get it right. The final example of therapeutic implication that I want to share is, I think, really the most satisfying to us, although in some ways also quite straightforward. Here, a young child presented with a progressive, very serious neurological disease. The clinical team had no idea what it was, but came to the view that it could possibly be an autoimmune disease and decided to treat aggressively on the basis of it possibly being an autoimmune disease. But because they weren't sure, they also asked us to sequence her and to do the analysis just as quickly as we could, since it seemed to them possible that we could get an answer that might inform about treatment options. We did that. When we sequenced her, we found that she is a compound heterozygote for loss-of-function mutations in a gene called SLC52A2. This is a quite intolerant gene, and moreover, one of the mutations that she carries has already been observed in a number of patients that have a very rare genetic disease. That disease is due to loss of function of this gene, and the other mutation that she has is a clear stop-gain mutation sitting right in the middle of the protein. So this genetic evidence really made quite clear that this child had this rare genetic disease. It turns out that SLC52A2 encodes a riboflavin transporter. The rare disease that it causes is Brown-Violetta von Lehr syndrome, specifically type 2. What is of particular importance here is that this is a very rare disease, but with a known treatment. For patients that have this disease, what you do is supplement their diet, in fact, with vitamin B. And it has been shown that the earlier the supplementation, the better the patient does. And in fact, patients can do very well if their diet is supplemented early enough, and the disease can be and probably usually is fatal otherwise. Having got this diagnosis, we of course immediately wanted to begin vitamin B supplementation for the patient immediately. And so we had a series of discussions with officials at Duke about how we could do that. This is a timeline for the study of this patient. Bloods were drawn on the family on day 1. After 20 days, we knew what she had, and we were quite confident. We communicated that to the clinical team. 4 days later, the patient came in to see the clinical team, and her mother actually gave her vitamin B in the lobby of the hospital that very day. And this is our patient. Her name is Kara Green. Pictures, of course, shown with complete permission. Before the initiation of treatment, Kara could not hold her head upright. She drooled frequently or constantly. And characteristic of this disease, she had extreme upper body weakness, was not able to lift her arms. Consistent with expectation for improvement when treatment is initiated early, her symptoms began to be reduced immediately upon administration of vitamin B. Head control improved, drooling was reduced, and really most striking of all, we saw her just a few weeks ago, several months after treatment was initiated, and I and some of the others involved met her with the cameras rolling, and she ran around the room high-fiving literally everybody in the room. And we all remembered when she could barely lift her arms. There really is no more job satisfaction than a geneticist could ask for than to see something like that. And to me, this really creates an absolutely overwhelming case for deployment of these kinds of genetic approaches in these kinds of patients, especially when we recognize that it is all but certain that what Kara really has would never have been determined without sequencing her exome. And the reason for that is that all the aggressive treatments that were being pursued really camouflaged what the underlying disease was. And it really makes you wonder how many other patients like that are out there. So with these kinds of experiences in mind, we have been thinking that it really is essential that we not only perform this kind of genetic analysis on as many patients as possible, but we do it as well as possible. And I do not mean any criticism directed towards any of the diagnostic labs that do this kind of work, but it is fair to say that the diagnostic labs that sequence exomes and genomes don't do the work in the same way as research laboratories are able to do it. And so with that in mind, we've been thinking about how to organize the data that are increasingly being generated clinically to improve the analysis of those data. And so in partnership with CURE, the epilepsy charity, and under the really remarkable leadership of Tracy Salazar, what we've been able to do is put together an initiative that will organize and consolidate into a single repository exome data for patients with epilepsy that's clinically generated. The basic way this works is that when a clinician orders an exome from, say, GeneDx or elsewhere for a patient, they will offer the patient family the opportunity to also enroll in a research study, which is the EGI, the Epilepsy Genetics Initiative. And if the family chooses to enroll, the lab will send the sequence data to the repository, and the people that manage the repository will undertake to reanalyze those data every 6 months. And so what that means, for example, is that if a new epilepsy gene is discovered, it will be possible to go through the repository and find out who has epilepsy because of that gene and communicate that information back. Now, in the first instance, we have a number of participating sites that will be enrolling patients into the EGI. They are listed here, but we are also in discussions to expand these sites, and in fact, Columbia University will be added because of our our upcoming move to Columbia, and there will be other sites added in as well. So that concludes the genetic stories that I wanted to tell you. I just want to emphasize that the progress really is quite remarkable, astounding in some ways, but it also does create some challenges for us. One of them is we absolutely must create clear standards for declaring genetic diagnoses. We also need to think about the speed of providing genetic diagnoses. The example of Kara really makes clear how important it is to get this information back quickly. And I also want to point out that examples like KCNT1, where there is an obvious functional model that you can use to see what the effect of the mutations are and to see what drugs might help, those are the minority. And so what I want to conclude with is just pointing out that if we're going to really have the full benefit of this progress we're making in genetics in terms of developing targeted treatments, we absolutely must develop a generalized functional modeling paradigm that can be applied to mutations found in any kind of protein, not just ion channels and so on. And specifically for epilepsy, we have persuaded ourselves, at least, that the multi-electrode array platform represents a really remarkable opportunity for general modeling on the bench top of mutations that cause epilepsies. And the basic approach here is that you've got electrodes sitting in wells. You grow neurons and some associated cells in those wells, and the neurons form neuronal networks with real synaptic connections. And over time, the neuronal network develops with firing patterns and synchronizes invariably. And you monitor the action potentials occurring in this neuronal network using the electrodes. And the idea is that if you have a mutant either introduced by genome editing, or if you take the neurons from a mouse mutant model, for example, if you have a mutant that creates a phenotype in this in vitro neuronal network, you can wash drugs through that neuronal network to find a drug that reverts that phenotype back towards wild type. And then that is a drug that you can try in a patient. And I want to just conclude by giving you an example that I think makes clear that this paradigm actually works. So in partnership with Wayne Frankel at the JAX, we have been working on a mouse model of his involving CEL4, which encodes an RNA binding protein. This is work being done by Yifan Liu in the lab. And so what we've done is take neurons from the CEL4 mutant mice. We have grown them in these wells with electrodes in them to monitor the behavior of the neuronal network. And what we see in these neuronal networks is a clear phenotype of the mutation. And so what's shown here is monitoring the neuronal network over time, and we show the number of spikes that occur in a so-called network spike. So this is when there's a whole bunch of neurons firing at the same time for the mutant in red and the wild type in black. And what you can see in all the experiments that we've run for neurons taken from multiple mice, we always see more spikes in network spikes for the mutant than for the wild type. And this actually can be viewed as an excitability phenotype that we see in this in vitro neuronal network. Now, most importantly, the work that Wayne had done had already suggested that there's a drug that might help in this type of epilepsy mediated by CEL4. And the idea is straightforward. Wayne was able to show that deficiency of this mRNA binding protein CEL4 had an effect on a serotonin receptor. And because of this effect of the mutants on the serotonin receptor levels, there was an indication that fluoxetine, which is Prozac encoding a selective serotonin reuptake inhibitor, might actually help. And so he gave fluoxetine to the mice and found that indeed the seizures associated with this mutant model were reduced by the administration of fluoxetine. So you know where this is going. We then wanted to test whether fluoxetine actually did anything in our in vitro neuronal network. And strikingly, what we see is a clear reversion of the mutant phenotype towards wild type when we introduce fluoxetine in this neuronal network. And so that's indicated here, where we see in the red circles that the knockout for cell 4, when you add fluoxetine, is pushed almost back to wild type. And what does that mean? What that means is that if we didn't have any idea about what kind of drug we might use, we could create this neuronal network, test out candidate drugs, and then see which of them works, and then bring them back to the patient. And the final comment, and this is absolutely anecdotal, who knows if this is real, but we have, of course, looked through our own genetic data to see whether we have any patients that have cell4 mutations that might be causing their epilepsy. Astoundingly, one of the patients that does have a cell4 mutation that might be contributing to their epilepsy, that patient actually was put on Prozac for 12 months for depression, obviously not for seizures. And that 12-month period was the only 12-month period for the recorded history of that patient in our clinic where he achieved seizure freedom, an indication that this really might be helping and showing how we can take this kind of an approach to really provide targeted therapeutic opportunities for patients. So that's where I will end this slide. I hope will acknowledge anyone that I failed to mention as we went through. I do want to emphasize in particular Vandana Shashi, a partner in all of the clinical genetics work that we've been doing, Mohammed Makati and Yangwei Zhang, who have also been partners in the clinical genetics work, and specifically who have been following the KCNT1 positive epilepsy patients and also the riboflavin case, Kara Green, along with Vandana Shashi. And thank you all for your attention.
Thank you, David, for this wonderful talk. Really inspiring stories of genetics and research in action. So just as a reminder for everybody, you can drop questions into the GoToWebinar control panel. And this talk is also recorded, so you can view it again later or share it with friends. So David, as we get into the questions here, first, kind of a technical one. You had mentioned early on about de novo mutations in trio analyses and how if these analyses are done well, it can result typically in just a few de novos per patient. We have an audience member who's saying, I usually get dozens. So what's the difference? What's the key to doing that well?
So what we do usually is use multi-sample, well, always use multi-sample calling, at least within the trio. This is really important because one thing that specifically catches is are variants that appear to be de novo, but that have a little bit of support for the presence of a variant in one of the parents, but not quite enough to call. When you use multi-sample calling, those are caught and those aren't judged to be putative de novo. In addition, after we have multi-sample calling and a variety of filters to try to identify the ones that really are de novo, and that includes a filter on the variant to reference. I apologize for this being too technical for some. That includes a filter on the variant to reference ratio. After all of that, we actually visually inspect the variants before we take them out to Sanger confirmation, because even with all of those filters, there are some things that upon visual inspection are obviously not real. And then when all of that is done, we normally have only a handful, 2, 3, 4 candidates that need to go for Sanger validation. And in fact, more recently, it's been usually closer to 2 than anything above that. So there are a variety of things that you need to do in order to get down to a manageable set that you move over to Sanger confirmation.
Thank you. I think that was very specific and helpful. Another thing that captured the audience's interest, and mine included, is the intolerance scoring. So one of the questions had to do with the cutoff. I think you had showed you're highlighting red versus blue using a 2% on either end. So where does that 2% come from? Have you considered more of a continuous measure to expand beyond that?
It's a great question. So in the figure, it was 2% that we were highlighting. What we did for the mutations in patients and controls using the integrated prioritization scheme. We used a cutoff of the 25th percentile. So we took the 4,000 plus genes that are at the 25th percentile as being the most intolerant in the human genome. And the reason that we did that is that when you look at already known genes that cause neurodevelopmental diseases, the vast majority of them are amongst the 4,000+ most intolerant genes. And so that seems a reasonable cutoff currently. What I do want to emphasize, though, longer term, so that's what we're doing right now, what I do want to emphasize longer term is that the way we're going to be doing this work is not by using a single intolerance score for the whole gene. The way we're going to be doing this work is to be using intolerance scores for parts of genes. Now, exactly how we divide genes up and to get their intolerance scores in each part is a work in progress, but that clearly is going to be how this is done. But right now, the cutoff specifically for neurodevelopmental diseases that we have found most useful is the 25th percentile. In other disease areas, you would have to use something different. And I should say, for anybody that wants these scores for any gene of interest, there's actually a website that you can Google and find called by Googling genic intolerance, and you can get the scores for any gene of interest.
Great, and you just stole my next question about the part genome, part gene analyses. There was an excellent talk along those lines at ASHG this year that was drawing on the new exome aggregation consortium data, where there are over 60,000 exomes worth of data, and the work was even done just using these reference populations and looking at, I believe, they were loss of function versus less dramatic variants. So great to hear that you're moving in that direction. Are you in fact thinking of updating or involved in updating your scoring based on those larger reference collections?
Yeah, so that's already happening, in fact. So there will be a version of the genic intolerance score that comes out using those data actually in the coming days. And the point that you are implicitly making is absolutely right. The larger the data set, the better our ability to construct useful scores for parts of genes. And there is no question that we will very soon have versions both of our approach and a very analogous approach that was independently developed at the Broad. We'll have versions of those approaches that are applied to parts of genes in a number of different ways, using, for example, protein domains to define the parts of genes, or simply using exons or other approaches to look at particular parts of genes.
Well, David, thank you. I see we have reached the end of our hour, so in deference to people's schedules, we'll thank you, and we'll cut it off there. And if there are further questions here, we'd be happy to follow up with you later and pass messages through to David as well. Thank you, everybody, for coming today.
Thank you, Ben.